Satish,
On behalf of PG and Research Department of Microbiology, we welcome Dr. S. Suresh, Associate Professor, Department of Biotechnology, FSH, SRM Ramapuram. He has completed his bachelor's and master's degree in biochemistry from University of Madras. He completed his PhD in industrial biotechnology from Anna University. His area of specialization on enzyme and food technology, biofuel and bioremediation. He has been a faculty coordinator for MANAV, conducted by the Project MANAV, the Human Atlas Initiative in September to October 2021. He served as a session chair for the poster presentation of 2 days virtual international conference, Chemistry for the Next Generation, organized by SSN College of Engineering. He is a coordinator of research and development, Faculty of Science and Humanities, SRM Ramapuram. He has been awarded with I2R International Teaching Excellence Award 2021 by International Institute of Organized Research and Young Scientist Award received from AICTE sponsored in international conference organized by the Department of Biotechnology in Pratyusha Engineering College. He is a resource person and served as a session chair for the Oral in National Conference organized by the Department of Chemistry in SSN College of Engineering and National Workshop held at Department of Biotechnology in D.J. Vaishnav College. He was a chairman of Research Scholar, AC College of Technology. He also delivered many lectures and expert talks on various topics. He planted a tree at Usaini Government School, Ramapuram. He is a member of American Chemical Society in Washington, Association of Chemistry Teacher, N. Purav Mark in Mumbai, Society for Advancement of Chemical Science and Education, IGCAR in Kalpakkam, the Indian Science Congress Association in Kolkata. It's our pleasure to have you with us, sir. Thank you.
Now, I request Dr. P. Vidya, Head Department of Microbiology, to welcome today's judge for paper presentation with a momentum. Thank you, ma'am. Now the Now the paper presentations will start. The first participant is Annapurna Sabanayakam from Hindustan Institute of Technology and Science. Can you guys hear me?
Good afternoon, everyone. I thank DJ Vaishnav College for giving me this wonderful opportunity. My name is Annapurna Sabanayagam, and my topic is about screening and prioritization of persistent, bioaccumulative, and toxic chemicals present in personal care products like toothpaste and talcum powder. So basically, personal care products are something which we use in day-to-day life, and which we have, it's one of the major consumables which we use all over the world globally. So obviously there'll be a higher production, and especially in India because we are one of the first populated country. So obviously we'll have a lot of consumables like shampoos, soaps, then toothpaste and talcum powder. So obviously we look into the chemicals that they use for producing talcum powder and toothpaste. So, what do you mean by personal care products is that it is one of the non-medicinal consumer products which the doctors won't prescribe most of them, and which we use for for admiring, for beautifying our appearances, or it could be exfoliating our dead skin cells, or cleansing, etc. So, what we're going to do is that we're going to look into all these chemicals or the ingredients that they use in each of the brands of talcum powder and the toothpaste, and then we look into all their what do you call is that they are known as canonical smiles, or simplified molecular input line entry system. So they are actually based in terms of they are represented, these chemical compounds are represented in terms of C, H and O. So they are in a simplified line entry form. So which these chemical compounds, they are these canonical smiles are available in PubChem. So from that we can find the canonical smiles, and then what we're going to do is that we're going to screen them. How we're going to screen them is that we're going to use the software program called EpiSuit, and in EpiSuit we're going to look into the how much persistence it takes, how much bioaccumulation it happens, and the toxicity level of all the chemical compounds. So first we look into the EpiSuit, which is actually developed by the US Environmental Protection Agency and Syracuse Research Corporation, and it is also freely available. It is a free software. So this is actually the front page of the episode, and you can see the input cas in which for each chemical compounds they'll have a number. So we'll copy the number, and then we'll take the canonical smiles from the PubChem which we have for each chemical compounds, and then we'll insert them. And also we'll input the chemical name of the compound, and then we'll look into the bioaccumulation, persistence, and toxicity. So the main objective of my study is that, so what we're going to do is that first we're going to identify the chemical ingredients of each of the talcum powder brands, and then toothpaste brands. And then next what we're going to do is that we're going to screen the organic chemical present in the toothpaste and talcum powder. Why we're going to take only the organic compounds is that inorganic compounds can be physically removed through sewage treatment. So when you're doing a treatment, the organic compounds are very minor and they cannot be removed physically. So that's why we're gonna screen the organic chemicals. Next, we're gonna prioritize the contaminants of the concern based on persistence, bioaccumulative, and toxicity using EpiSuit software. So the main scope of the study is that we can look into the emerging contaminants, or this or the identification of emerging contaminants. So if that chemical compound has the ability to persist and accumulate into the environment, then it is a hazardous waste into the environment. So those are known as the potential emerging contaminants. And then the personal care products are the main source of emerging contaminants that arise from the anthropogenic use on a daily basis. So anthropogenic use is that the contaminants which done by the human activity we get. So from this we can establish a list of prioritized chemicals that are potential of emerging contaminants of concern. And due to excessive and everyday usage of PCPs, the personal care products, they can be considered in for toothpaste and talcum powder. So this is my work plan. So first we're going to collect all the chemical ingredients from all the brands, and then we're going to categorize them into toothpaste and talcum powder. And then we're going to screen the organic compounds. We're going to selectively take only the organic compounds, and then we'll look into the episode software, and then we'll look into the persistence, bioaccumulation, toxicity. So before we go into the persistence, or what persistence or bioaccumulative toxicity is that, for example, a chemical compound is there. We'll go through series of it. So a chemical compound, if it is not biodegradable, then it is highly chance that it will persist into the environment. If there is a long persistence, then obviously they live for a longer period of time. If there is a longer period of time, there is a chance that they will accumulate into the environment for a longer period of time. If there is accumulation, then therefore they mean they are toxic to the nature. Thank you. So, just go back to the next slide. So, according to the REACH regulations the persistence of the chemical compound should be that means the half-life of the chemical compound, if it's more than or equal to 900 hours, then it is persistent. And bioaccumulative, if it is concentration factor is more than or equal to 2000 liter per kg wet weight, it is bioaccumulative. And toxicity, if the lethal concentration 50 is 96 hours, then it is toxic. So you can look into the gray areas. Those are the organic compounds for the toothpaste. And next one is the talcum powder. All the gray compounds are the organic compounds. If you look into the gray ones, they are actually only the toxic level that have higher bioaccumulation. They have high toxicity, and the primary degradation is that they will. That means the parent product will be broken down, and the ultimate biodegradation is that they will be broken down into carbon dioxide and water. So if you look into that, you can see days and weeks and months are done. So it will take a longer period of time. And you can look into the organic of the toothpaste. You can see triclosan and tocopherol acetate are the main hazardous chemical compounds. So the conclusion of my project is that so for the talcum powder, it is actually the two chemical compounds are that they are triclosan and tocopherol acetate from the toothpaste, and the talcum powder is isomethyl ionone triclosan butylate hydroxytoluene. And the prioritized chemicals are that means they are emerging contaminants, and therefore major steps to be taken like green synthesis should be taken necessarily to reduce the chemical compounds into the environment.
Okay. Any questions from audience?
Any queries?
Well, your presentation was good, and you were okay. Please go to the topic slide. So your screening and all the software one.
Yes, sir.
So in that, and you were taking the measurement from organic, only for organic.
Yes, sir. Only for the organic compounds.
Okay. The organic compounds, supposed to be you also choosing different brand, right?
Yes, sir.
So different brand, you're supposed to be choosing different brand. The source of each brand is supposed to be one brand may use for flower.
Yes, sir.
One they can use any liquid, the solid. Okay, they actually the sources are different.
Yes, sir.
And based on the sources, the organic has organic compounds what you choosing. So that also different.
Yes, sir.
So right. So in that area, No, what are you coming to say in this?
What I'm saying is that from all, like from the toothpaste and talcum powder, it is one of our personal care products. So obviously, the industries which produce the talcum powder and the toothpaste, the effluents do have the organic compounds as well. So even in their effluents, there are high chances that they do not degrade into the environment, and they will have higher persistence and bioaccumulation.
See, if you do, if you're taking the organic, particularly if you're choosing the inorganic things, whatever you justify, that is good. But you're taking the organic means obviously, the organic are degradable only, right? The organic molecule.
There are some chemicals that do not degrade, sir. Like triclosan, they do not degrade. So we are able to identify through this software.
Okay, that and all okay. Because it's green.
Yeah, yes, I understand. Because of the organic compounds, they do get degraded. Some of them, they are volatile, so they do.
See, time is actually in, okay, suppose choosing for organic things, they can, they can definitely degrade, but the time is may change. Okay, in having, so some compound may degrade over 3 days, some compound they may degrade, the time has to be changed. Okay, anyway, and you are choosing, the parameters. How you to fix the range.
I didn't get you, sir.
Actually, to fixing the some range, right?
Yes, sir.
Okay, how you can take it from?
So according to the REACH regulations, that is how the norms are.
Oh.
So from that, we got to know that once you're, taking them as waste, they should not have more. For the persistence, the half life should be less than 900 hours. The half life itself should be 900 hours below. Okay, so if their half life is more than 900, then that means there is a chance that they will accumulate into the environment.
Okay, good. Because your project is like that's it. All the best.
Thank you, sir.
The next participant is Mariamavit Gurmamilki and Renuka R from Hindustan Institute of Technology, followed by M. Esther Joyce and M. Sneha from Hindustan Institute of Technology. Please be ready.
Good afternoon, all. I'm Renuka, and this is my friend Mariam. And our topic is on the microbial degradation of waste. Our entire project was done on basically 4 different steps. The first one was the collection of samples, and the samples we used for the waste were from landfills. And the second step was isolation of the microorganisms from the samples we obtained. The third step was about the characterization of the obtained microbes after culturing process. And the last step was about the calculating of the degradation properties of these microbes on plastics. So plastics we generally know are inorganic polymers, and conventionally plastic is degraded by two methods. One is incineration, the other is by dumping in landfills. Now both of these methods have their own drawbacks. Now incineration method is to be carried out at like 1500 degrees Celsius, and these facilities are not available in everywhere. And the waste, the ashes that are deposited after this incineration process cannot be further processed. So these ashes are finally again to be dumped. Now the landfills, these cause a very long time of degradation, and also they reduce the soil fertility over time. Now this leaves us with another option, which is the biodegradation of these plastics. Conventionally, it is done using enzymatic processes where the polymers are broken down into monomers by enzymatic digestion, and then either anaerobic or aerobic metabolism is carried out by microorganisms. Now, in our project, what we aim to do was to use these microbes to culture these microbes which are capable of degrading plastics, even the non-biodegradable ones. So the method we used was the media we used was the enriched media along with polyethylene plastic strips, and for the culturing of the microbes we used nutrient agar. Now, the reason we used enrichment media for the emulsification process here was because the enriched media does not have an energy source of its own. The only energy source that we provided was the polyethylene strips. So, the only way for these microbes to grow was by the degradation of these plastic strips. This shows the degradation property of these microbes that we isolated. The first step was the soil collection. The sample collected, as I said, was from landfills across the city, like from areas of Perugudi and Taramani and all that. These landfills waste were collected. The second process was about subculturing. We isolated these microorganisms and we cultured them using the nutrient agar at 37 degrees Celsius for a few days. And after that, screening procedure was done, in which we took out subsequent passes. The colonies that were identified were categorized based on the morphological differences, and individual colonies were selected and they were further subcultured. Following this isolation process, we did for the screening, in which we used 150 ml of enriched media and we added these low weight polyethylene strips. The final step was identification, which is still in process. We are planning to use 16S RDNA sequencing method for this process.
Okay, so this is how we plated it. So we collected samples from three places and we named them as sample A, B, C. So first we cultured here, and then we isolated the colonies, and then for the unique colonies and well isolated colonies. Here is our enriched media. So here we added 0.1 gram of polyethylene strips, and then we took readings every 7 days. So actually the experiment lasted for 14 days. So we took reading the first 7 days and the next 7 days. So this is the result we got for the liquid screening using liquid media. So during the first week, we saw a drastic change on sample C, which the plastic the plastic reduced its size to 0.05 grams, which is 50%. And then for the next 7 days, some of them did show change in the plastic weight, but then another drastic change we saw was on sample B. Both of them showed like 50% of degradation. So it shows that the microorganisms found in sample B and sample C had a high capacity of degrading the polyethylene strips. We also did using solid media. For solid media, we used PEG as our plastic. So we did see some colonies emerge after 7 days. Like Renuka said, this project is ongoing, so we have to keep it for another 7 days to give a result. But then, since we did not see any inhibition on it. So for the results, like I said, like we did find bacteria that had the capacity to degrade the plastics and then for the solid media, we can't give a result yet since the experiment was not done. So our future scope is first taking the reading from the solid media, whether we find results or not, and then second one is going for 16S RDNA sequencing for the microorganisms we found in sample B and C, and also we will be doing some biochemical characterization, and also we do plan on taking this experiment to large scale production, so we will also be doing some studies on this. So to conclude this presentation, so we know plastic is a major concern in this world because of the usage is increasing, but then we don't know how to actually dispose it. So in this experiment, we did find microorganisms that can reduce the weight of low molecular polyethylene plastic strips, and also we did see microorganisms that are able to grow on PEG. And also we will be, for our further studies, we'll be cloning and doing an expression of plastic degrade genes so that we can apply this on larger scale. Thank you. And does anyone have any questions? Any queries?
Okay, well, which degree are you pursuing now?
We take biotechnology third year.
B.Tech Biotech, third year.
Yes.
Okay, so because for the presentation of international conferences, you need to move research. So because that's initiated by you, right? So we need no, more research because it doesn't think what micro, right? So and actually only plastic also the consuming rate is 50% is means so we're choosing for bacteria. It is bacteria can consume any kind of which we using any kind of synthetic it consume because the food particularly if you look for the plastic they they take as nitrogen or carbon sources. They are kind of unique perspective. Anyway, my best wishes. Thank you.
Thank you, sir.
The third participant is M. Esther Joyce and M. Sneha from Hindustan Institute of Technology, followed by Ms. V. Satyasini from Dwarakadas Govardhandas Vaishnav College. Please be ready.
Hello, am I audible? Okay. So, good afternoon, everyone. This is Sneha Muralidharan and M. Esther Joyce. We are from Hindustan Institute, and we'll be presenting the topic production of bioplastic from agricultural waste. Our guide for this project was Dr. R. Anita, and moving on, we have plastic is painful for the environment. Plastic pollution is one of the leading environmental concerns that we are facing, and since it is polluting vast our ocean, soil, and causing a lot of landfill problems as well, it is our major focus for our study. Yes, so that this would be production of bioplastic from agricultural waste would be our topic. Next slide. So for today, we'll be covering the topics like just an overview of the present case scenario of what the bioplastic is and the agricultural alternative that we are using, as well as our objective and more information about our studies. Next slide. So this is the present case scenario, and plastic waste is changing the so-called blue planet to plastic planet, and it stands that more than 300 million tons of plastic waste is being produced extensively across the globe, and this graph was taken from an article published in the PubMed. Next slide, please. So bioplastic. Bioplastic is basically a form of plastic produced from biopolymers. So by biopolymers, we mean it is extracted from biological sources. It can be microbiological or plant sources as well. So bioplastic has two components: the biopolymer and a plasticizer. The biopolymer would be like PLA, that is polylactic acid, or PHA, that is polyhydroxyalkanoates. And for our study, the plasticizer we used was glycerol, and we aim that bioplastic to be an innovative step towards a circular economy, by which we mean that it will aims to keep raw materials in a closed loop. Next slide, please. So we are focusing on agricultural waste as an alternative source. So more than approximately 20% of domestic waste is food waste, and all of these wastes are high in hemicellulose and lignin as well, which can be extracted to form biopolymers like PLA and PHA before mentioned. And one of our samples, next slide, please. Yeah, this is our aim. So our samples include gelatin, banana peel, orange peel, tapioca starch, cassava, and potato starch. So basically, bioplastic can be classified as starch-based, cellulose-based, and protein-based. So banana peel, cassava starch, potato, that would be coming under starch-based, where the biopolymer would be PLA, polylactic acid, and gelatin would come under protein-based, and the protein would be gelatin, of course. And moving on. This is the existing research that has been done on this topic. So we have production of bioplastic from orange peel as well as from gelatin waste. Coming forward, we have the principle that will be taken over by my co-presenter.
So on moving with, so next slide. So we'll move on with the principles. So this is what we had. So first of all, we just collected the raw materials, so which is nothing but the gelatin, the proteins, and also the agricultural raw materials. So subsequently, like what are the raw materials we select? That's in the first stage. And second stage, we just extract the starches from it. Like we do the process of grinding, and the next other process of sieving, and we just do blending it, and then we just dry it, and the finally what is this is what our plan, like what we did in our project. So what are the additives we add here is like water, glycerol, and like NaOH. These are the things. Next one. So as well, these are the features what we noticed. Like it is recyclable, and it is a renewable, and it is non-toxic. So even we just we did the toxic test. So it is non-toxic, and it is biodegradable, and it reduces the carbon footprint. Next one. So these are the main thing. These are the visible results what we got. So these are they are under characterization and development technique. So this is an ongoing project. The above top corner one is a bioplastic from cassava what we obtained. That has gone for the tensile and FTIR, SEM, temp test, all these results. This is a bioplastic from cornstarch. Next. This is bioplastic from banana peel what we obtained, and this is bioplastic from potato starch. So gelatin, bioplastic from gelatin was our main core objective. Like we just visited the soft gel company, we just brought the capsule waste, and from that capsule waste, actually collagen is already present in that capsule waste. So if that is a protein, so this is protein based bioplastic. So we just made it, and we also found the degradation. So all these things are under the development and characterization. So this project of us is the development and characterization of the bioplastic from agricultural and industrial waste. So the next slide. Next, if Gavin, let's see.
So our study basically focuses on the quality and degradability of the bioplastic from eco-friendly sources and the effect of the biopolymer at different concentrations. So with solid waste in the form of various plastics accumulating all over the world, bioplastic is an important source of innovation, and the creation of newer bioplastic is always seeking out new resources. Next slide, please. Future perspective, it is being applied in a lot of biomedical industries for scaffolds, tissue engineering, bone reconstruction, and packaging as well. Next slide. Conclusion, replacing conventional plastics is a new avenue for sustainability, as conventional plastics uh, and bioplastic share similar properties. Next, we have an awareness video. Next slide, please. Any questions?
Okay, Maria, right? Your name?
Yes.
Sneha. Okay, Sneha. So your presentation is good. The thing is, you people also let this say. So your initial work only doesn't work. Okay, you went an international conference. Okay, anyway, what is my question? What is the major difference between synthetic plastic and bio?
So synthetic plastic, we have the polymers that are non-degradable, as in they have high persistence and all. Like, and for the bioplastic,
No, no, no. I'm not asking no the nature of so chemical formation, the structure of plastic and bio.
Sir, actually, that petroleum-based plastics, this chemical structure,
So
it's
which
being
means
fermented and then converted into lactic acid and then into a polylactic acid. So this would be the
so which means hydrocarbons are more having a synthetic plastic,
yeah.
whereas bioplastic have the
It's
more.
it's less greenhouse gas.
So what you term that bioplastic, that is PHA. Okay, have you heard about the PHB?
Polyhydroxybutyrate.
Okay.
This is.
So you also when you go for agricultural waste, no, uh, that is more tedious process I think. So when you go for you know that is you can extract from microorganisms easily. You also yielding also high when compared to the other waste. Because anyway you are going to use extraction process, particularly using for organic solvent. Okay, that is also that is not eco-friendly. So, and that the initial process, and you also using glycerol, sodium hydroxide. Okay, then what is the pH of sodium hydroxide? Glycerol, why you are using that?
Glycerol we use it since it's a plasticizer, so that's why.
Okay.
But that we just added like according to the consumption, but more like we add it only for since it's a plasticizer. That's
So what is the pH if you are supposed to be using sodium hydroxide?
it's actually the alkaline level. It's below 7.
Below 7.
So,
Okay, yes, thank you.
okay.
All the best.
Thank you.
Participant number 4, Ms. V. Satyasini from Dwarakadas Govardhandas Vaishnav College. The fifth participant will be Sundar Blessy Sara from Madras Christian College. Please be ready.
Namaste. Good evening to one and all present here. I'm Satyasini, Research Scholar, Department of Chemistry, PG Vaishnav College, going to present on phytoengineered silver nanocubes and platinum nanospheres using mixed aqueous extracts of Platycladus orientalis and Sapindus mucorosea. It's like chemically synthesized metal nanoparticles introduces harmful substances to the environment, and it is also affects the health of the living organisms. Nowadays, green synthesis of metal nanoparticles is using plant extracts is gaining more importance because of its eco-friendly nature, and it's also cost-effective. Next slide. Here we have used two plants. Number one, Platycladus orientalis, which is a Chinese herb. In common man term, we can call it as puja. And number two is the Sapindus mucorosea. It is actually our Rita or soap net that we use along with sugar to wash our hair. And these two plants are authenticated from CSMCARI, that is Ministry of Ayush, Government of Tamil Nadu, before we start our work. Next slide, please. First, we prepared the aqueous extracts. We didn't use any organic solvents or even alcohol we didn't use. We just used the water as a solvent. So we prepared the aqueous extract of plant products. For that, the plant products are washed using deionized water and shade dried for 10 days and just blended in a common kitchen blender and stored it in an airtight container separately. Then for the preparation of Platycladus orientalis, 0.6 gram of it is taken in a 20 ml of deionized water, soaked it for half an hour and stirred it at 60 to 70 degrees Celsius for half an hour, then soaked it overnight as it is. In the case of Sapindus mucorosea, 6 gram of it is taken along with 20 ml of hard deionized water, soaked it as it is for overnight. Then in both the cases, in the next morning, we just filtered it using a 20 micron sieve and centrifuged it for 10 minutes in a common clinical centrifuge. Then the supernatant solution is collected, which is used as the plant extract and is a reducing agent to reduce the metal ions from the metal solution. Next slide. Next slide. Modus operandi. In the case of preparation of silver nano cubes, we have taken 95% of silver nitrate along with 5% of plant extract mixture, stirred at 90 degrees Celsius for 2 hours to get the silver nano cubes. In the case of platinum nanoparticles, we have used the same methodology, but the time duration is increased because the platinum is somewhat resistant to reduce. And we have used the one variable change at one time method to fix the parameters like time, temperature, concentration, extract percentage, extract ratio, mixing ratio, etc. Next slide. Characterizations. UV spectrum of plant extracts. It is C is actually Cucumin platycladi. That is a bright form of Platycladus orientalis, and P is SC is actually the Sapindus mucorosea extract. Both the extract shows peak at 290 nanometer. It is because of the N to pi star transition of carbonyl group that is present along with the phenolic groups. That is actually the reducing agent present in the plant extract. And the peak at around 350 nanometer is because of the flavonoids that is present in the plant extracts. Next slide. UV spectrum of silver nanoparticles. The left spectrum shows the formation of silver nanoparticles. The LSPR peak at 490 nanometer for 3 different temperatures have been taken, around 30 degrees Celsius, around 60 degrees Celsius, around 90 degrees Celsius. That 90 degrees Celsius fellow is the efficient fellow showing the high intense peak and the blue shifted peak. So it indicates the efficient formation of silver nanoparticles, and the right spectrum shows the formation of silver nanoparticles at the same 420 nanometer at different time intervals from 60 minutes to 180 minutes. UV spectrum of platinum nanoparticles, it doesn't show any characterized LSPR peak. it shows only the peak of plant extract. Sometimes it will show the continuum from 300 nanometer to 500 nanometer. It's because what's the section I have is it doesn't have any free electrons like silver or gold. IR spectra of plant extracts. POE, Platycladus orientalis extract, and Sapindus mucorosy extract. Both show strong, broad peak at 3,300 centimeter inverse. It's because of the OH stretching of the phenolic groups present in the plant extracts, and the peak at 1,600 centimeter inverse is because of the carbonyl stretching of the carboxylic acids present, and the characterized peak which is present alone in the Sapindus mucorosy extract is because of the at 1045 centimeter inverse is because of the CMOS stretching of the carbenols that is present in the sulfonin content, which is a natural surfactant present in the Sapindus mucorosy that is responsible for the reduction and also for the capping of the nanoparticles. IR spectrum of silver nanoparticles and platinum nanoparticles. Here you might have known that peak is somewhat reduced in the silver nanoparticle, that is the peak at 1042 centimeter inverse. And also in the case of platinum nanoparticles, the peak is not there at all. XRD of silver nanoparticles. Here we are using the mixed aqueous extracts. That means we have tried many ratios of both the extracts, that is 1:0, 1:3, 1:1, 3:1, 0:1, like that. But the better performing fellows are 1:3 and 1:1. And for that we have taken the XRD. Both the 1:3 and 1:1 shows the formation of crystalline silver nanoparticles, especially the structural space and the so same image of silver nanoparticles. 1:3 shows more nanospheres than nanocubes. In the case of 1:1, they are giving nanocubes, more nanocubes than nanospheres. Next, please. Same images of platinum nanoparticles. I have taken for 1:3 ratio plant extract produced platinum nanoparticle that is showing some aggregation and also it gives some nanospheres. Results and discussion, that is what I have discussed by seeing all those spectras. But I want to stress one thing: the polyphenolic groups that is present in the plant extracts of the common reducing agents, that is reducing the metal ions from the solution to metal nanoparticles, and the capping agents are actually the carbonyl groups, the oxygen delta negative and delta positive will be the metal nanoparticles. And thanks for the opportunity. Thank you so much. Any questions?
Any queries?
Well, basically you take two plants. So actually you confirm the metal and okay whatever choosing the particularly plant. Both you
Yes, yes.
so both you collecting from these two, right?
Yes.
Kana
Precursor is actually silver nitrate solution, sir, for silver, and for platinum it is hexachloroplatinic acid. That is a precursor we have taken, and the reducing agent are the plant extracts. Modus operandi one.
I'm actually the okay, which one is platinum, which one is silver?
So this one is plant extract units.
Okay.
Next. And this one is both are silver, sir. Okay. This is taken for different temperatures, and that is for different time intervals. And this next one will be the platinum. This one is the platinum. It shows only the plant extract peak, sir. It doesn't show any at all, because there is no localized surface plasmon resonance for platinum nanoparticles according to the literatures.
So what my question is, you're choosing different parameters for silver nanoparticle because
Yes.
different temperature and different.
Yes.
What about that? Doesn't show any
Same parameters we have tried for platinum also, sir.
So
That gave me more headache than silver, because At the beginning, I was so enthusiastic choosing platinum because it doesn't show any localized surface plasmon, so I have chosen it. But the precursor what I choose is hexachloroplatinum acid. Instead of that, if I take platinum, potassium containing platinum salt, that will be more easier to reduce. That platinic acid is resisting to reduce.
That is your research actually. See, everyone can do the plant extract, particularly on the children, a particular food, they can extract. But if you're that is because you are the researcher now, so you can challenge yourself, and you can work out on the mechanism. So yeah, so quantitative mechanism. You can try. And so next up, so this here the eye aspect of plant extract here is showing the because you're talking about the plant extract, functional group.
Yes. And the next slide, the IR spectrum. This is a IR spectrum of silver nanoparticles and platinum nanoparticles.
From which plant?
Sir?
From
both
which
the plants.
plant?
Both the plants we
Both
have
the
used.
plants.
Yes.
So only one thing. That's right.
So 1:1 ratio. This one is a 1:1 ratio. It is mentioned here for silver nanoparticles. I have used 1:1 ratio of plant extract. That means 20 ml of reaction I'm taking 20 ml of reaction content I'm taking. This 19 ml will be. Thank you so much.
Okay, good.
Sir, thank you so much. Thank you, sir.
The next participant is Sundar Lessi Sara from Madras Christian College, followed by Ritika Debroy from Belur Institute of Technology. All the participants are requested to maintain silence in the auditorium, and also please don't use mobile phones. Thank you.
Good afternoon, everyone. I thank DJ Vaishnav College for giving me this opportunity to share my study. I did the study under the guidance of Dr. P. Veda ma'am. So, my study is based on the antibacterial activity and cytotoxicity properties of the medicinal plants, Pandanus armeniferous and Cissus quadrumularis. So, I was more interested in this both plants because they're most commonly found, and the Pandanus armeniferous plant is also called as pandan plant. And most of us love biryani, so we can find this plant, the plant in biryani. So, slide. So coming to the introduction, so in these days we take antibiotics very frequently. So even for a small headache, we take antibiotics. So our body has become resistant to the antibiotics. So we are in need to research. We are in need to find the antibiotic resistance drugs. So we use plants. We use medicinal plants to derive the medicinal drugs. So that's for the reason I choose Pandanus ambifolius and Cissus quadrangulosus. So these both have very good medicinal qualities. So the Pandanus ambifolius plant is used as anti-diabetic tonic from in Ayurveda, and in Cissus quadrangulosus is used for or treating obesity and bone diseases. It is also called as heteros plant. So it is also used for treating fractures and all. So the aim of my study is to study the antibacterial activity, phytochemical analysis, and cytotoxicity properties of the taken medicinal plants, materials from the leaf extracts. So the Ayurveda has confirmed that the leaf extract contains many components which are useful for the medicinal uses. So the objectives of my study is to first the collection of leaves from both the plants. So the fresh and disease-free leaves were collected, and the second one was the preparation of leaf extracts. So we use acetone, chloroform, ethanol, and water. And the next one is to check the antibacterial activity of the extract. And the next one was to determine the phytochemical analysis of the extract. And the final one was the cytotoxicity properties of the prepared extracts. Next one. So coming to the collection of the plants, as I said, the leaves should be pure and disease free. And after collecting, they were washed with distilled water and they were shade dried and they were powdered. And after filtration, they were preserved in a dry and clean container. So as I said, methanol, ethanol, acetone, and chloroform and water were used to prepare the plant extracts. So these are the plant extracts. So coming to the antibacterial activity, we did by the well diffusion method. We Muller Hinter was prepared and sterilized, and they were poured into sterile petri trays, and they were waited until they solidified. And we took three test organisms from the stock culture, namely Pseudomonas, Staphylococcus aureus, and E. coli. And using a sterile swab, we swabbed each of the petri plates, and 5 wells of 5 millimeter diameter were punch, and 1 ml of different extract were added into the wells, and they were incubated at 37 degrees for 24 hours. So we will that. So coming to the phytochemical analysis, so to test for the phenols and tannins, 2 ml of 2% ferric chloride and solution was added to the extract. A blue-green to black color indicates the presence of phenols and tannins. So in the Pandanus amerifolis plants, we can see alkaloids, which shows antiviral properties, and so they are used to treat for herpes virus and influenza virus. So coming to the test for flavonoids, crude extract was mixed with 2 ml of 2% sodium hydroxide solution. An intense yellow color was formed, and it turns to colorless solution, and a few drops of dilute acid was added, which indicates the presence of flavonoids. Coming to the test of steroids, crude extract was mixed with 2 ml chloroform and concentrated H2SO4, and a red color produced. The lower ring was chloroform layer indicate the presence of steroids. So these are the tests for phenols and tannins. So phenols and tannins were both present in both plants. So coming to the flavonoids, the solution turned into colorless. So the presence of flavonoids was found in Pandanus amerifolis, whereas in scissors there was in acetone we didn't find it. Coming to the steroid, there was no presence of steroids were found in pandan plant. So coming to the cytotoxicity effect of the cell line, so the cytotoxicity of the cell sample was tested against A54 cell line of adenocarcinoma lung cancer, and it was collected from Pune, and it was tested by MTT bromide assay, which was found by T. Mossman in the year 1983. The cells were seeded in the 96 well microplates and incubated at 37 degrees for 48 hours in 5% CO2 incubator and allowed to grow at 70 to 80% confluence. Then the medium was okay. Coming to the results, so we found the antibacterial activity of this plant extract show the sensitivity to the gram-positive Staphylococcus aureus. Both the plants were sensitive to gram-positive organisms and resistance against the gram-negative organisms. So coming to the cytotoxicity assay done for Panda summaris, we took ethanol and methanol plant extract because the alcoholic extract showed more antioxidants. So when the control was when the concentration was low, the cell viability was high. So when the control was 24 mul, the cell viability was 93.36, which confirms the cytotoxicity of the. In the next slide. The next slide. So we can see the spectrophotometry results. Next slide. And these are the results of the methanolic extract of pandan plant. So yeah. So I like to conclude by saying that the leaves of the Pandanus amelifolis plant contains flavonoids, which increases the because of that the cytotoxicity levels was noted, and yeah, thank you. These are the references. Yeah. Thank you. Any queries?
So the blessing. The study is antibacterial activity and the cytotoxic properties of medicinal we are choosing. Good. So in that antibacterial study, why are you choosing gram positive and also 2 4?
There's three organisms.
Okay.
Staphylococcus, Pseudomonas.
Okay. Which one you know? Why that particular three organisms?
Stop.
So, cytotoxicity test.
So MTT bromide acid.
Cytotoxicity.
A549 cell lines.
No. Edoda cell line.
So adenocarcinoma lung.
Lung cancer.
Lung cancer.
Good. All the best.
The next participant is Ritika Debroy from Vellore Institute of Technology.
Am I audible? So good evening everyone present here. My name is Ritika Debroy. I am a PhD research scholar working under the guidance of Dr. Sudaramaya ma'am, who is a professor in Department of Biosciences, Vellore Institute of Technology. And today I'll be presenting my research work entitled Novel Curcumin Derivative Can Interfere with Mursi Ligase Dependent Peptidoglycan Biosynthesis in Multi-Resistant Salmonella type B. And we have used in silico approach to look for alternative therapeutics against REST type B because in silico approaches have always been proven to be fast and efficient methods to screen out novel molecules against disease complications. So, next slide. So, a very quick overview of the research problem I want to discuss here is that we all know that S-typh is the causative agent of enteric typhoid fever, and recently the main problem in treating the complication is that the emergence of multidrug resistant S-typh. And we can see over the last 3 decades, whichever antibiotics have been prescribed over these years, S-typh has emerged resistant strains against each of the antibiotics. Hence, looking for alternative therapeutics against S-typh has become a need of our right now, and hence this research was performed to look for phytocompounds as therapeutic solutions, because we know that phytocompounds have very low to none side effects, have immense wide range of beneficiary properties, as well as they have shown multi-target specificity, that is, its ability to target more than one protein at the same time. Hence, phytocompounds were chosen as potential solutions, potential therapeutics against mitigating this complication. Next slide. So this is the first of all we needed to shortlist the protein targets. We have gone through literature and we have found a comparative genomic study where they have prioritized 19 major drug targets, 19 major vital proteins in S-typh, and they have told that as potential drug targets in the organism. So what we have used that information, and we have defined the protein structures, as in we have reduced the Ramachandran outliers, and we have reduced the local and gross errors in the protein structures, and we have reduced the internal energy of the protein structure to get the most stable structures out of it. And side by side, we have screened the ligands. For getting the list of phytocompounds, we needed to have a reference molecule, so we have used curcumin as the reference molecule. Curcumin can be found from turmeric. It has already reported antibacterial, antifungal, anti-cancer activities. So curcumin derivatives were used to screen for alternative therapeutics. So its pharmacokinetic screening was done, and the parameters were Lipinski violations, GI absorption, and ease of synthesis of the compounds were taken as parameters for shortlisting the compounds, as well as it should have safe toxicity profile. So 16 compounds were finally screened out with safe toxicity profiles and screened out pharmacokinetic properties. Next slide. So we have gone for binding affinity assessment and we have taken binding energy as the parameter. Binding energy is the least amount of energy required by the ligand to bind with the protein. So if the binding energy is less, it means that the ligand can easily bind with the protein structure. Right? So we have compared, which all the molecules are basically curcumin derivatives, and we have compared the binding affinity with curcumin and 4 prescribed anti-typhi drugs. So we can see here the highlighted ones with the red ones are they are the 6 curcumin derivatives which have shown high binding affinity towards more than one protein, towards multiple proteins as compared to antibiotics. So these ligands were considered as multi-specific ligands. And another important point we have interpreted from this plot is that all these six multi-target specific ligands have shown the highest binding affinity towards the same protein, and that is muciligase protein, which is involved in peptidoglycan biosynthesis of salmonella type. If we go next slide. So this is a zoomed out version of the previous thing. Is that we have only this binding energy shown for only the Morse protein. So we can see here is that those 6 bars are showing they are the multi target specific ligands, and we can see that they are having higher binding energy than the antibiotics. Most importantly, another inference that we have drawn is that molecule 8 have shown the least binding energy against Morse. So we have taken this combination, this molecule 8, that is a deoxytetradeutero derivative, and Morse protein as model combination for studying the docking for going for the docking studies. Yeah, so before going to docking studies, we have gone for site-specific docking. For site-specific docking, we need to identify the active site first. Because if we have to functionally inhibit the protein, we have to identify the functional domain so that the ligand can bind there. So we have identified the active site there, that is the ATP binding site of Morse protein, where ATP is the natural substrate of the protein. And we have these are the residues in the that functional domain, and we have checked for the dynamicity and the flexibility in the C alpha backbone as well as the RMS values in that protein as well. So this is the molecular docking studies, and this is the site-specific molecular docking. That is, the ligand is bound to the active site, that is the functional domain of the Morse protein. So we have compared. First one is the curcumin derivative, second one is the curcumin, and the third one is the ATP, that is the natural substrate of the protein. And there are most of the residues were overlapping with each other. Another point that we have seen here is the binding energy graph. Here we can see that the 3 bars are with Morse. First one is curcumin derivative, next is curcumin and ATP, and the binding energies were compared with that of standard anti-typhic compounds as docked with the respective protein targets. And hence we can see that the deoxytetradeutero derivative has shown the least binding energy or the most binding affinity, and it can be a competitive inhibitor for the ATP binding site in Morse protein. Yeah, so the results were validated with the RMSF fluctuations, and we have seen that in presence of ligand, there has not been much differences in the RMSF values. So these are the main conclusion that this combination, this curcumin derivative with Morse protein, can be an ideal combination for designing anti-typhic therapeutics, and yeah, so we could publish the research work. So yeah, I would thank Vellore Institute of Technology for motivating me to do my research, my PhD guide, and Professor Anand Barasu has been there, has given the lecture, and my lab mates who have been literally nothing less than my family. And last but not the least, I would thank DG Vaishnav College for giving us the platform to share our research. Thank you.
Yeah, your presentation was coming. So, what is my question? Choosing curcumin, right? So, in from curcumin, we extract some novel.
No, sir, we have not done the extraction part. The entire work was in silico only. So we have gone server based, and we have just given the structure of curcumin, and it was server based analogs.
Yeah, analogs you can.
Yes.
Okay. And that multi-drug resistant, particularly on the salmonella typhi.
Yes.
Okay. So, from your result, what I got that, particularly on going for the ligand, the choosing ligand, particularly on 6 ligand molecule that
Yes.
bind onto the
Yes, yes.
like this. Yeah, thank you. All the best.
There is a little change in the slot. The next member will be Aman Kumar Jaishwal.
A very good evening to one and all present here, respected judges, and all of the students who are sitting here to watch me present. So, the topic of this conference is Next Generation Microbiologists, Challenges and Opportunities. So, what I thought was I would present a topic that would actually be about the opportunity. It was interesting listening from the morning to the various perspectives of the resource persons and other presenters. So that's why I have decided that I would be presenting my review paper titled Bridging the Gap Between Astrobiology and Paleobiology, biological mechanisms of the organism, and to understand the effects of solar radiation as well. When we try to understand these two topics, we'll come to hypothesize if there is a possibility that during the earlier years of the Earth, interplanetary transport of microorganisms took place in any. Next slide. Okay, so coming to the various experiments that have been conducted previously by other researchers, we know that factors which affect growth in the atmosphere uh, are the first is the solar radiation, because we know our atmosphere, Earth's atmosphere has many layers: the troposphere, stratosphere, mesosphere, thermosphere, exosphere. So it has layers, and in the outermost layers of the atmosphere, the concentration of UVB and UVC is so extremely high. Even 30 kilometers above sea level, it cannot support life. Similarly, even 29 kilometers above sea level, temperatures are so low it mimics the temperature of that of Mars surface. And obviously, because the atmosphere thins down as the height increases, there is unavailability of nutrients and gas molecules to transfer these nutrients to microbes. But in 1975, there was a breakthrough report that reported that Serratia marcescens could undergo cellular division, while in a nutrient containing aqueous droplet about 2 to 6 micrometers in diameter. It was assumed that microbes cannot grow in air because the air is not a good supporting medium because it does not contain any nutrients. But in 1975, it was reported that Serratia could in fact undergo cellular division when uh, when media was presented as a form of aqueous droplet, even in minute size, that is 2 to 6 micrometer droplet, the Serratia was able to utilize the nutrients and divide. Next slide, please. And further studies in the late 1900s reported that in the outermost layers of the atmosphere, Staphylococcus, Micrococcus, and Deinococcus species can be found. So they were found from the upper troposphere and the lower stratosphere layers of the atmosphere. Even further studies revealed that pigmented bacteria were found in the stratosphere and troposphere, which was about 5 to 20 kilometers above sea level. Similarly, they were also found above 77 kilometers in sea level. These are three studies conducted in 1967, 1978, and 2006 by different researchers. So they found that even about 20 kilometers, 77 kilometers, you can find microbes. Similarly, four species of bacillus were isolated 24, 28, and 41 kilometers above sea level in India with the help of a hot air balloon. Yeah, so stress, we know because outer space is a lot of elements that can produce stress to the organism. When stress occurs, what we usually do is we'll get angry and we'll get up and leave, right? So, but microbes can't do that. They have to survive and they have to adapt to the stressful conditions. So, in the form of microgravity, Pollard in 1965 was the first to suggest that microgravity will produces no effects on cell more than 10 micrometers in diameter. So because most bacterial cells are below 10 microns, he proposed that microgravity would not produce an effect on these organisms. But further studies reported that microgravity in fact did change the growth and behavior of the organism. It reduced lag phase and increased final cell population. The hypothesis behind this is because microgravity um, interfered with the environment of the cell, it interfered with the nutrient uptake of the cell, due to which the cell had to adapt by inducing a new biological response which gave different results such as reduced lag phase and increased cell population. And non-model organisms were more susceptible to this because non-model organisms, there is less mass transfer between a cell and surroundings if it is non-motile. A motile organism, if it moves, then there is some form of energy transfer between the cell and the environment. But in non-motile organisms, this does not take place. So they are more susceptible to um, the effects of microgravity because of the reduced mass transfer during weightlessness, that is during microgravity. Next slide, please. Another stress factor is radiation. We know that radiation affects the proteins and nucleic acids. It is directly absorbed by these proteins and nucleic acids, which leads to cell damage. And it was first tested, the effects of radiation was first tested by Hornick on two organisms, Bacillus subtilis and Deinococcus radiodurans. Both these organisms showed double-stranded breaks, but they were quickly repaired by homologous recombination or non-homologous end joining, two mechanisms of repair. And he concluded that radiation was low enough to not kill the cell, but high enough to increase mutations. By the way, all these experiments are carried out by simulating outer space conditions. They are carried out by they are carried out in an apparatus called a wall bioreactor, which uh, simulates the gravity of outer space as well as high particle accelerators, which simulate the radiation which is produced by the cosmic rays. So, I wanted to conclude that since some organisms were actually reported to survive this experiment, there is a possibility that during the earliest formation years of the year, these microorganisms were transported by interplanetary measures, that is meteorites, entrapped in the meteorite in the form of spores, and could lead to the rise of, could lead to a new theory which gives the theory of life on Earth.
Yeah, very good, Amar. For the topic itself is something actually. Okay. Actually, I learned a few things that is new information that now the atmosphere with macro, each organism from sea level to the atmospheric, how they were actually from the nutrient how they consuming their growth, particularly. Very good, good work.
Thank you, sir.
For the
The next participant is B. Aishwarya, Apollo Arts and Science College. All the participants are requested to skip the introduction part and continue with the rest of the slides. Thank you.
So, good evening, everyone. Myself, Dr. B. Aishwarya, I'm an assistant professor in the Department of Microbiology, Apollo Arts and Science College. I'm here to present my part of the PhD work. Was done under the guidance of Dr. K. Vijay Ramesh, assistant professor in the Department of Plant Biology and Biotechnology in Qaide Millat Government College for Women. So my research work entitled Novel Endophytic Fungal Metabolite, Promising Candidate for Drug Resistance Reversal in Clinical Microorganisms. Antimicrobial resistance poses a well-organized global health threat due to the global dissemination of bacteria to the antibiotics resistant. An antibiotic consumption proves the bacteria also becoming the resistant to those antibiotics.
This is the scenario of global aggregate resistance to E. coli, Staphylococcus aureus, Klebsiella species to third generation antibiotics. And this is the E. coli resistance alone to the third generation antibiotics. Next slide. Hence, we scoped our research, overall research objectives for the. From the seaweeds, we isolate the endophytic fungal strains and bioactive preliminary screening, and then morphological identification. Then to the optimization of secondary metabolites, characterization, and to the study of antimicrobial resistance mechanism. In this presentation, we are about to see the characterization study and antimicrobial resistance mechanisms. The proposed methodology for the purification of metabolite usually we carried out in thin layer chromatography, silica gel column chromatography, HPLC. For the bioactivity, we did for TLC bioautography method and MIC FIC analysis. For the cytotoxicity of metabolite, we did the MGT assay. So for the characterization of fractioned metabolites, we go through the UV spectroscopy, FTIO spectroscopy, GCMS and NMR. Next slide. For the antimicrobial resistance mechanism, the in vivo and in silico studies were carried out. In the in vivo study, we screened for the 3 resistance mechanisms. So one of those in cell wall membrane disruption and the efflux pump inhibition study and the beta-lactamase enzyme inactivation study. So these are the clinical drug resistance bacterial species: Staphylococcus, beta-lactamase, MRSA, ESBL Escherichia coli, MDR Pseudomonas aeruginosa. So these strains are procured from Sri Ramachandra Medical College and Research Center, Porur, Chennai. Next slide. This slide shows the isolated strain of Penicillium daliae from the seaweed Enteromorpha flexosa, which is collected from the Covelong sea source. So that is the microscopic images of EF and molecular conformation image. Next slide. So the extracted crude metabolites were initially purified by the thin layer chromatography. So those spots are not visible in the iodine chamber, and then we visualized in the UV mode. Then the sample at the RF value 0.55, the fluorescent bands are visible. So then the column eluted fractions were carried out, and then the purified fraction were quality checked by HPLC. So these are the images of TLC bioautography method of TLC plates against the multi-drug resistance organisms. Then apart from the TLC bioautography method, the minimum inhibitory concentration, fractional inhibitory concentration of crude metabolites and column eluted fractions were analyzed from combination with different antibiotics. For gram-positive bacteria, we checked with penicillin G. For gram-negative bacteria, we tested with tetracycline chloramphenicol, polymyxin B antibiotics with the combination. In that tetracycline, here 4 fractionated extracts show synergistic with tetracycline antibiotics. So this is the cytotoxicity assessment of the fractionated extracts of Penicillium daliae. In normal vero cell line, we tested the extracts at 1 mg per ml with the higher concentration. So this image shows the normal viability. About 90 to 92% of viable cells are visible even at the higher concentration. That reveal the extracts are not toxic at the concentration. Next one. So in the characterization study, we carried out in UV visible spectra and FTIR, and by NMR. In GCMS, all the retention peaks show the same compound up to decadienal chloride. So from the FTIR and UV spectra studies, we come to the end the EF extracts may be a fatty acid moiety, but in the NMR it will show us the structure named as then hydroxy dimethyl andecyl tetrahydropyranol, which is named as a penicillin. This compound is till 2019 unreported in India. Then for the study of antimicrobial resistance factor, we have to go through the different mechanisms.
Thank you. So, slide here. So actually, it was an endophytic fungal metabolite.
Yes,
So, this, can you tell me the name of that?
Fungi. Penicillium gallicum.
So, what my question is, in the HPLC, because end of the research you showed that actually the characterization studies are shows fatty acids.
Yes, fatty acid moiety in FTR. And then in MI.
And the NMR shows that structural similarity. So, no, it actually comes.
Actually, GCMS will only taken for the fatty acid compounds like only.
Which means you're using the solvent reactor, the extraction of this particular molecule, you're using the fatty acid. Okay, that solvent only used. When supposed to be going to the NMR, that character
shows the different carbon atoms.
So what is the conclusion? So
in end of the slide, we like to confirm that we completed the drug resistance mechanisms. About the stated 3 mechanisms are completed in the laboratory methods that cannot be explained with the
and for the HPLC, FTE, so the prediction should be accurate manner.
Yes, sir.
So please concentrate on because the characterizations are important. When you go for the drug-based thing, so characterization is very very important. Each and every functional group plays the match. Yeah. So, yeah. Thank you. Good. All the best.
Participants, I repeat, please skip the introduction part and continue with the rest. Now, it's S. Kavya and team from Professor Dhanapalan College of Science and Management. Good evening to you and all present here. I am Kavya from Professor Dhanapalan College of Science and Management. Good evening, sir. Now, my topic is applications of ozone treatment and natural ingredients in kombucha. Prevents to prevent the side effects caused by the opportunical bacterial strain. Kombucha. Next slide. Kombucha is anyone? Okay, sir. Kombucha is a fizzy, sweet, and short drink
It is made with tea. The kombucha is a traditional drink in China. It prevents the side effects like hair loss. It's many sorry, health benefits. These are the health benefits, especially it prevents the joint pain, weight loss, and booster energy. Next slide. Side effects. Kombucha is safe, but it mixes unspatulated and with different type of bacteria and yeast. To the bacteria lead the infection in some peoples. Next slide. Methodology. These are the ingredients we use for the experiment. Plain water, tea powder, jaggery, yeast, lychee juice. Next slide. Preparation of tea extract. Now prepare the tea extract by using the jaggery powder in instant sugar. Next slide. Fermentation process. First, we prepare the 600 ml of tea extract by dividing the 5 ml of 10 jars. Now we prepare the 4 controls and first 2 controls we add the tea extract and then 2 controls we add the plain water. Testing. Testing we add the ingredients, natural ingredients and added yeast and lychee juice and jaggery. Next slide. Ozone treatment. The ozone treatment, national ingredients in 3 jars and we labeled in ozone 5 minutes, 15 minutes and 13 minutes. The fermentation process in 10 days. And the ozone treatment is daily to observe the color changes and next slide. Results and discussion. Make the observation is the present fermentation process in fruity alcohol smell. The ozone treatment samples, untreated sample is spoilage. The ozone treated sample is not spoilage. How to identify this? The methylene blue staining is used for the ozone treatment sample. Only the yeast is observed. Next. Estimation of alcohol by hydrometer. How to identify the alcohol level? We use the hydrometer, the ozone treatment sample. First, ozone 5 minutes is 30 percentage of alcohol. Then ozone 50 minutes and 50 percentage of alcohol. The ozone 30 minutes and 50 percentage of alcohol. The specific gravity. Next. Conclusion. We conclude the drink should be dilute in water. Consuming is better result. Thank you.
Okay, Kavya.
Yes, sir.
Your பேரை மட்டும் kombucha.
Kombuchas.
Okay, fine. So in the kombucha, so what is the major ingredient?
Kombucha is சைனாவோட ட்ரெடிஷ்னல் சார். அவங்க வந்து ஸ்டாஃப் ஆன்டிஃபங்கல் ஏஜென்ட்டுக்கு வந்து ஸ்டாஃப் வேரியூஸ் யூஸ் பண்ணியிருக்காங்க. அப்போ ஸ்டாஃப் வேரியூஸ் யூஸ் பண்ணும்போது சம் சைட் எஃபெக்ட்ஸ் வந்து காஸ் பண்ணுது பீப்புள். அதனால நாங்கள் வந்து நேஷனல் இன்க்ரீடியன்ஸான டர்மரிக், பெப்பர், க்ளோஸ் அதெல்லாம் யூஸ் பண்ணுவோம். ஓகே சார். தேங்க்யூ சார்.
The 10th participant is Mahalakshmi S&T from Hindustan Institute of Technology and Science. All the participants in the auditorium are requested to maintain silence.
Good morning to everyone present here. I'm Monica, this is Mahan, this is Hema. So we'll be presenting about the categorization of pesticides based on persistence time. Right, so pesticides are defined as chemical substances used to destroy, prevent, repel, or mitigate pests ranging from insects, microorganisms, weeds, and rodents.
So recently there was a pesticide management bill that was passed in 2020, and it was to regulate the import, manufacture, export, storage, and quality and the use of pesticides. But nothing was written about the chemical properties, the fate, the transport, and environmental effects of the pesticides. So in the view that pesticides will have a detrimental effect on the environment, uh, this study aims to create a readily available database with information such as persistence time. Next slide. Before that. So, an important factor that will affect the degradation time of pesticides is the persistence time. So, persistence time is basically measured. It is basically the half life, which is the rate at which the compound breaks down to half of its initial concentration. So, for estimating the half life, we have an EPI software to screen the chemicals. So, I'd like to repeat again, the main aim of this study is to create a readily available database that has information about the compounds, persistence time, and because this is an ongoing research, we'll also give toxicity and biodegradability of the pesticides.
Yeah, I am sure like you have read some of the information about the EPA software. One of us, as discussed by one of my seniors. Now let me explain in detailed way. The estimation program software is nothing but which is a Windows-based software which is used to find the physiochemical properties. The methodology that we have followed for the estimation of pesticides and the categorization is first we have taken the approved pesticide list by the Central Insecticides Board, and then we have obtained the canonical smiles from the website called PubChem. And then chemical smiles is nothing but which is used to translate the three-dimensional structure of pesticides into a string of symbols, which can be easily understood by the software. And then the EPA through the help of EPA suite software, we have estimated the persistence time, and with the help of persistence time, we have categorized into high persistence, medium persistence, and low persistence with a specific range of values. And these are the pesticides. There are totally 39 insecticides, and the fungicide and bactericide are 28, and the herbicide are 4. And the organic chemicals are 17, which is 9 microorganisms based and 6 are micro plus chemical based. And there are totally 87 pesticides in the list as of now approved by the Central Insecticide.
So, according to the CIBRC, Central Insecticide Board and Research Committee, they approved 87 chemicals which can be used for our agriculture purpose. So, for the next slide. For our easy representation, for easy identification, we gave some color representation, which the blue indicates the high persistent chemicals, and the orange indicates medium, and the green will indicate the low persistent. So if you see Actara which has the persistent days which is about 73. The normal high persistent period, maximum high persistent period for a chemical is 60 days, but it almost exceeds 73. Can you improve? And there is a pesticide name which is called Metadol, which has more than 360 days, which is also called a bioaccumulation, which is harmful to our environment. So, overall, out of 87 approved chemicals, 40 chemicals are highly persistent. Using UPI suite bioinformatic tool, we found the results. The main concern towards this topic is about the chemical persistent time and off-life period. So this gives us the information about how pesticide acts towards our environment. Biomagnification is a real-time problem where accumulation of chemical which affects our whole food cycle and will reduce the life expectancy of every organism. So to overcome this problem, we can introduce some efficient biopesticides and bioinsecticides.
Well, categorization of pesticides process. Destination was good. The thing is, you people also using that piece of So this is also like software related project. Good. All the best.
The 11th participant is Deepa Rekha from Government Arts College for Men.
Good evening to one and all present here. I am Deepa Rekha from Government Arts College. My guide is Dr. K.P. Girivarajan. My title is Bioprospecting the Fungal Communities of Some Algal Species for the Production of L. aspergillus, which is an anti-cancer drug. Yes. So briefly, I am describing about the L. aspergillus. So it is a neoplastic drug. It is an anti-neoplastic drug which acts against which stops the blood circulation in neoplastic slugs. Basically, it was extracted from bacterial cells, but it causes allergic reactions to few patients. So scientists have been focusing on other multicellular organisms like fungi. Particularly, I am focusing on endophytic fungi for the production of the same L. aspergillus. So my study site is Kovalam, and I have collected three algal species, namely Gracilaria, Gratilupia, and Ketomorpha, from the intertidal zones of Kovalam Beach. And the samples were processed within 24 hours from the time of collection in the laboratory. So for the isolation of epiphytic fungi, I followed Gunasegre needles dilution, serial dilution plating method, and for isolation of endophytic fungi, I followed Dobronic methods. And for the production of L. aspergillus, the Zebeck-Stokes medium amended with 3% phenol, red phenolic reagent, which is an indicator, has been used. The incubation time for epiphytic fungi is 7 days, and for endophytic fungi it is 4 days. 12 to 12 photo period has been followed. Next. So what are the results which we have found in the research have been analyzed using softwares. The colonization frequency, relative percentage of occurrence of groups of fungi, and the species diversity index, which is the Fisher's alpha, which have been calculated using the biodiversity flow software, and coefficient of similarity have been calculated. So we have isolated 12 species of fungi, which includes both epiphytes and endophytes. Of these, 4 were epiphytes and 10 were endophytes. And the species Ketomorpha yield no endophytes. Only one epiphyte has been isolated from Ketomorpha. Next. So the Jacquard similarity index shows the very less similarity or more dissimilarity between the epiphytic and endophytic fungi. And the Fisher's alpha index shows species richness and evenness in Gratilopia when compared to Gracilaria. And the hypomycetes group of fungi dominated the overall fungal colonies which were being isolated, and the species diversity were found more in Gratilupia when compared to Gracilaria. Next. So in the plate assay, Zebeck assay, 10 species were taken for production of L. aspergillus, out of which Nodulisporium and Chetomium produced L. aspergillus in the plate assay. The pink zone which is formed around the inoculum shows the presence of L. aspergillus. Next. So I'd like to conclude that we have taken 3 species of algae, namely Gracilaria, Gratilupia, and Ketomorpha, for screening epiphytes as well as endophytes, of which 12 species have been isolated from the algal thalate taken for studies, of which 4 were epiphytes and 10 were endophytes. Oleobasidium and Cladosporium have been found in both epiphyte as well as endophyte. In Ketomorpha, there have been no endophytes. This could be due to the less volume of the thalate structure. And in Gracilaria and Gratilupia, there is a gelatinous cavity in which the endophytic fungi or epiphytic fungi could able to trap, but it is not present in Ketomorpha. This could be a reason that there were no endophytes isolated from the Ketomorpha. And on the L-asparagines plate assay, the nodulosporium and cladosporium produces L-asparagines. So this study shows that endophytic fungi are a scope to extract novel metabolites, particularly as L-asparagines, which is used in lymphoblastic leukemia, can be isolated from the endophytic fungi. Thank you.
Actually, the aim for this project is to production of large batches, right?
Yes.
For the using of anti.
So,
For no application is to be anti-cancer.
yes, sir. Particularly lymphoblastic leukemia.
Okay.
It is currently in use, but the source is from bacterial cells. So we are focusing, but it causes, as I told, causes some allergic reactions to some patients. So research have been focused on other microorganisms. So I have taken endophytic fungi and it produces L-asparagines. So this could be an alternative.
So asparagines, what type of catalytic reaction is that?
So
Asparagines is what kind of catalytic reaction?
it is an anti-neoplastic drug and it alters the blood flow in neoplastic cells and it makes the cells starve, lacking nutrition, and ultimately the neoplastic cells will die.
Okay. All the best.
Thank you.
And the last participant will be Priyamvada from Vellore Institute of Technology.
Good evening, everyone. I'm Priyamvada, working under the kind supervision of Dr. Sudharamaya ma'am. She is a professor at SBST, VIT Vellore. Currently, today I'm going to talk about the computational tools and which is being employed in the discovery of new drugs and discovering new antimicrobial resistant genes. Those tools I have accumulated all together here. What is K pathogens actually, and why AMR is important to study? AMR is something called antimicrobial resistance, which is prevailing all around the world. It is a global concern because there is no antibiotics left to deal with it. Antimicrobial resistance, the third generation of antibiotics are there, which is also not working out, such as cephalosporin is there, Enterobacter is not working on it. So there is this computational biology bioinformatics approach can help in dealing with to identify the antimicrobial resistant gene. K pathogens was first being identified by named by the IDSA, Infectious Diseases of Society of America, where it named it as Enterobacter faecium and Staphylococcus, Klebsiella pneumoniae, Enterobacter baumannii, Pseudomonas aeruginosa, Enterobacter species. These all bacteria are actually available in the hospital also. So the patients which are going into hospital are, even if they are cured of any kind of surgery, cancer or anything, they are more prone to these bacterial infections and that can even die of it. So there is a need of identification of new genes which is being prevalent over here. So this is the stats taken from WHO, which is showing that AMR is going to peak up to 10 million cases in 2050 till 2050, and especially you can see this Asian countries is showing the most resistance being shown by the bacterial species present in Asian countries. Next slide, please. So, while shown here that what WHO says about AMR, it is a global concern because of its reoccurrence. And GLASS also says that fluoroquinolone resistance is widely shown by 70 to 80%. And this is the mechanism which is already discussed by our professor, Professor Anand sir. And but still little bit is there in how these antibiotics are not workable for these bacteria. This is the complete how this bacteria releases some of the enzymes called beta lactamases, where it breaks the beta lactam ring, and even they can alter the peptidoglycan layer, such as penicillin binding protein, which changes into penicillin binding protein 2A, and it is less shows less affinity to the beta lactam antibiotics. Like this is the main where we have discussed about the in silico approaches, where we have discussed about the genomic approaches, systems biology, and structural biology approaches. In genomics, there are various approaches such as whole genome sequencing. We can do transcriptomic analysis. We can metagenomic studies we can perform. In systems biology, where these two identities, you can see there are two words called as degree and one is edges. Degree, we call it as the protein, any kind of gene or any kind of metabolite, we treat it as a degree. Where there is an interaction between these two protein or genes, we call it as edges. This is the various tools and various tools are employed in the genomic approaches, where we can do the whole genome analysis just to check the AMR determinants or SNPs, or you can check for any kind of mutational studies you can perform. These are the various tools which is already employed already. So accumulated all together, it will be beneficial for the students to, if they want to carry on the studies. Next. In systems biology approach, where we talk about the interaction, how in a particular cell, the particular two gene actually don't physically attract. Protein only physically interaction occurs between proteins. So there are protein-protein interaction databases where you can accumulate all the there is 14,000 organisms in a string database, where you can check the interaction between them. And for host pathogen interaction, there are other databases where you can go and get the interaction, and you can check the functional annotation of those genes. There are several gene ontology terms called as molecular function. You can check on biological processes, and through gene regulating network, you can check the transcription factor to activate or inactivate a particular protein. You can check through how a transcription factor goes and bind to a gene and it activates. Those things you can study there. And in structural biology approaches, where you once you get the protein of interest, where it is showing that okay fine, we have to work with, we have to check on go on for the structural analysis. In that we go for protein optimization, mutational study about that protein. For protein optimization, we do minimization of the energy, and the structural refinement we predict the secondary structure, tertiary structure. And at last you can end up with, like, after doing the structural biology, like once you get the protein into your hand, you can go on for your lead compounds to dock with it and get the therapeutic marker as well as your therapeutic leads. So this is all what we have concluded from how we can end up into identification of the marker till the discovery of the new lead compounds. This work has already been published in World Journal of Microbiology. And thank you all. Thank you all for giving an opportunity, especially the organizer of DG. These are a team working there.
Okay, good. Actually, this is for review. Okay.
Yeah.
Okay. So in the silicon, in silico approaches, particularly on combat with any AMR,
Yes.
only we can go with pathogens or any other leading material supposed to be proteins, any biomolecule.
Any biomolecules, metabolites, proteins, genes, anything. So it is already these kind of studies we are doing in cancer biology and all. But escape pathogen, it is the need of ourselves, so it is good to work with it.
So that escape pathogens. Okay. What is the major significance of this? What is the major significance when you're using for this escape pathogen?
The amount of investment, like when you do whole genome sequencing, you can get new variants, and you can identify where the particular change in the protein sequence it is happening, or how it is like peptidoglycan, they are changing the peptidoglycan protein. So you can study those where the change is, and then you can design the drug according to it. It is like site specific, you are getting more into it. and it is like
So it is, very simply said, it is very specific to the site. Right?
yeah, like in COVID, if we would have dependent on the conventional, we would have stayed there only. But now when we used the computational tool, it paced the work, and so we got the vaccine.
So if supposed to be site specific, particularly when you go for protein and we have a different bond, hydrophobic, hydrophobic.
Yeah, interactions.
Okay, so the structures also varies, right?
Yes,
So,
yes.
so that and also we can predict and
Everything, sir.
everything we can predict.
Till there, from starting to end, how to deal with it.
Okay, from precursor molecule to the end of molecule. Yeah, very good. Thank you. All the best.
And there is one last participant from Professor Dhanapalan College of Science and Management. It's R. Niveta.
Ma'am, 1 minute.
Good evening to one and all present here. I'm Niveta from Professor Dhanapalan College of Arts and Science Management. My topic is herbal treatment against NDV in poultry. Ranicot disease or NDV is severe disease that caused to the birds or in the domestic poultry. Caused to the poultry birds, and it is infected birds are treated with vaccine for prevention. Instead of vaccine, we use turmeric and garlic, pepper, and jeera in this treatment. And these are the clinical manifestation: and twisting of neck, paralysis of wing, facial edema, diarrhea, and drop in production. These are some of the clinical manifestation. And we use garlic, pepper, turmeric, and jeera, and cooked rice. We mix it together, and cooked rice is used for the binding agent. And these are feed to the infected birds. First 3 days, we can see some changes in the birds, and after a week we can see the complete cure in this. Instead of vaccine, we can use these bioactive compounds for the NDV treatment. We can identify the infection by the swabs, vocal swabs, and facial edemas in the birds, and it can be treated according to the identification. As the finally, we are concluding that pepper, turmeric, garlic are most important bioactive compound we can use instead of vaccine. Some of importance of the turmeric, and turmeric is used as an antiviral and antibacterial activity which destroys and spikes the virus. So, எங்களோட பவுல்ட்ரியில் இருக்கிற பேர்டே எடுத்து அதுக்காக நாங்கள் ட்ரீட்மெண்ட் பண்ணி பார்த்தோம் சார். அது இப்போ நாங்கள் இதை கம்ப்ளீட் பண்ணல சார். இப்போ ஃபர்தராக ஜென்ரலாக நாங்கள் இதை வச்சு this is new cattle disease virus. Cattle disease virus. Yes, sir. Cattle virus disease. Cattle disease virus.
I, on behalf of PG and Research Department of Microbiology, would like to thank our judge, Dr. S. Suresh, Associate Professor, Department of Biotechnology, SRM, for taking out time from his busy schedule. Your time and dedication made this event possible, sir. Thank you very much, sir. The results for today's paper presentation will be announced tomorrow in the valedictory session. I thank all the participants for joining us today. Meet you all tomorrow. The event will start sharply at 9:30. Thank you.